What you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. Hey everyone, I want to share with you a new AI-powered research tool I've been using to find scientific papers and learn from the literature. It's called Consensus. It is an AI-powered search tool built using large language models trained to help you find and understand the best science faster. Consensus helps you not only find individual papers, but its AI models will help you assess research quality and even synthesize insights across multiple papers for you. You can ask it general questions about any topic you might have, such as what are the known risks and benefits of a certain prescription drug? What do we know about how a certain food or nutrient affects the human body? Anything you can think of. Its Pro Analysis feature will actually provide you with a concise written summary of what the literature says, broken down point by point, highlighting the strength of the evidence across papers and providing you with individual studies ranked by the relevance and including additional information like citation rates, journal quality, and more. You can also save, cite, and share individual studies with a single click. Unlike other AI and search tools like ChatGPT, PubMed, or Google Scholar, Consensus uses over 20 custom large language models that are developed and trained for this specific purpose. The base Consensus product is totally free, but the premium product that you can subscribe to has better features and unlimited queries. And I highly recommend this if you are using outdated tools like PubMed or Google Scholar, or you just do your own research. For a limited time, you can use the code MINDMATTERSPECIAL, all one word, to get a free premium subscription for 1 year. That is a $150 value for free if you use the code MINDMATTERSPECIAL today. So click the link in the episode description or check my support page and give Consensus a try. Use the code MINDMATTERSPECIAL to unlock your free premium subscription today. Intro for yourself.
Yeah, so my name is Jibran. I am an associate professor and Canada Research Chair in Translational Neuropsychopharmacology at Western University in London, Ontario. And I am a neuropharmacologist by training. Did my PhD at the University of Toronto, followed by a postdoctoral fellowship at Dartmouth College in psychiatry, and then started my first research program at the University of Guelph down the road. And now I've been at Western for 2.5 years. The focus in the lab is around the co-occurrence of substance use and serious mental illness. And we're interested in substances across the board, from alcohol to nicotine to cannabis. And then we, as far as psychiatric conditions go, we dabble in schizophrenia, bipolar disorder, and even some work in ADHD now. And we're really interested in cracking the chicken or egg question, in that there are relationships that go in both directions when it comes to substance use and psychopathology. And so we're really interested in trying to crack that.
Yeah. And I think we're going to probably spend a lot of time talking about the co-occurrence and the relationship between substance use disorders and certain mental illnesses. But, stepping back for a moment, even more broadly, there's a lot of comorbidities between mental illnesses, substance use disorders, all of these types of things. A sort of meta result in the broad field of psychiatry and mental illness is that, even though we have a DSM-4 that categorizes, all of these illnesses into discrete names and categories, we've got depressive disorder, we've got anxiety disorders, we've got schizophrenia. We have words and labels for all these things, and it's very easy to talk about them as islands, as these separate things. But as you know, and as many people know, but not everyone appreciates, these things co-occur at a very, very high rate, much, much, much more than you would expect by chance, such that for almost any mental illness you can think of, if you have schizophrenia or an anxiety disorder or a substance use disorder, what have you, your odds of having some other mental disorders, usually multiple, are much higher than they are for the average person in the population. Can you just talk about that a little bit and how you start to think about that and unpack that as a neuroscientist?
Yeah, so, let's think about it in a few different ways. There's only one brain, and the brain is very much a concerted network of networks that act together. And so if there's a pull in one place, there's going to have something's going to give in another place. And so we need to understand that we don't have neat bins in the brain that you can file things into. It's not like Inside Out, right? Like it's messy when it comes to the brain. And so I think it makes sense that there would be co-occurrence. There's genetic overlap between a lot of conditions, both in the psychiatric space and the substance use disorder space. But in addition to that, there is network and brain connection and brain area overlap. The same brain region, the mesocortical limbic dopamine circuit that we always talk about it with substance use, also comes up with schizophrenia. And so that there's that brain region overlap. But important that you mentioned the DSM-5, right? Like DSM-5 is only really good for charging codes for insurance, right? Like comorbidity is the rule and not the exception, right? And even when it comes to substance use, polysubstance use is the rule and not the exception. And even when you look at diagnostic overlap, right? When you look at across the different diagnoses, if you take each of those symptoms that make up the DSM-5, right? Like there's like 600 something symptoms that make up the DSM-5. Now, how many of those repeat across multiple diagnoses? I think over 200, 230 symptoms repeat over 1,000 times between those diagnoses. So it's not as if that now that we created these neat little windows or neat little bins and pockets, that there wasn't no overlap between them. In fact, the overlap even within the DSM is the rule and not the exception. So I think that probably captures a lot of the different levels where the comorbidity can occur.
Yeah, it's not as if there's a circuit in the brain that breaks with schizophrenia and a separate circuit that breaks only with anxiety and so on and so forth. And even for things like substance use, it's not like there's one circuit completely dedicated to nicotine and one completely dedicated to opioids and so on and so forth.
And it's because substance use isn't just one thing either. Even when you think about the addiction cycle, the euphoria is something else. And then the withdrawal and negative affect is something else. And the acute craving and the preoccupation is something else. And those are already three distinct domains with three different parts of the brain and very different outcomes and phenotypes. So really, I think it would be far too reductionist to try to reduce any of these things down to one thing.
And I think maybe we can just start by talking about schizophrenia, because schizophrenia ties into many different related topics that we can spend quite a bit of time on. So there's the illness itself, which is very debilitating, and we can talk about. It's a serious mental illness. It's not not to belittle anything else, but depression is often mild. It can be very severe, but schizophrenia is, if you have it, it's typically quite debilitating and can be completely debilitating. And then so there's the neurology and neuroscience to talk about, but also, as we'll come to, there are sex differences with schizophrenia, differences between males and females in terms of how it manifests. And when it manifests, there are developmental effects. It tends to arise at certain times in the lifespan. And then there's comorbidities, especially when it comes to substance use disorders. So schizophrenia is sort of an interesting single illness to talk about because it can also relate to all these other domains that are interesting. But let's just start very basic here. What is schizophrenia, and what is sort of the core? What's the core of schizophrenia in terms of what's going wrong in the brain?
Yeah, so before I get into it, schizophrenia is also probably not just one thing, right? So we'll talk
Mm,
about what schizophrenia can generally be, but
yeah.
it's probably a spectrum as well, a psychosis spectrum disorder of
Yeah,
sorts.
yeah. Would you say that's pretty common? Like we talk about schizophrenia or we talk about depression, but each of these things are probably some number of distinct illnesses that we we
Collapse
often
it.
can't tell apart because their sort of endpoints are similar.
Yeah, and especially when you think about bipolar disorder with psychosis, where does bipolar disorder with psychosis end and schizophrenia begin? There's probably not a clear line that
Mhm,
a lot of people can draw, right? So
mhm.
I think it's important to know that there are overlapping boundaries probably, and that those sorry about that uh,
No worries.
sound. Muted. Um, I think there is probably an important thing to consider in terms of those overlapping boundaries.
Yeah.
But yeah, so schizophrenia is a serious mental illness, as you said, very debilitating serious mental illness that affects nearly 1% of the global population. Um, the three primary domains in which we see symptoms are positive symptoms, negative symptoms, and then cognitive symptoms. And this is not that there are symptoms that are good and symptoms that are bad.
Yeah.
Positive here means something that is added that wasn't there before. Negative is something that was taken away that was there before. And cognitive symptoms would be related to cognitive dysfunctions. And so what is being added is now hallucinations they might be auditory, they might be visual, they might be paranoia, there might be so those are the domains that we
Yeah.
usually see,
Yeah.
and those are
You're
in the
you're hearing voices
psychosis.
or seeing things. Things are
Yeah,
being added to your perception that
yeah,
would not normally be there.
would not have been there. And then what's being taken away is feelings of pleasure. So anhedonia is a symptom
Mm,
in schizophrenia where you don't feel pleasure anymore. You lose your ability to connect socially with others. So social deficits and dysfunctions also arise and can be considered a negative symptom. Mood, again, as we were saying, non-overlapping boundaries. Mood is also significantly affected. And then in the third space, that's the cognitive deficits, cognitive dysfunctions. That's where you see a loss of working memory, changes in ability to pay attention, changes in ability to, navigate complex tasks. But in addition to that, even at a more simpler level, things like sensory motor gating, like how does your brain respond to sensory stimuli? And you can understand that even in the positive symptom side, there are things that we usually respond to sensory stimuli, what we hear, what we see, but then our responses shift and they change. And so they're the cognitive domains, and you can again, even within each of these domains, one can affect the other. If you're not understanding the world around you in the same way as you were, it then makes it that much harder for you to navigate it or communicate with it or right, so the negative symptoms and the cognitive symptoms might overlap, or your now positive symptoms are now affecting your ability to hear the person that is speaking to you right in front of you. So that might affect your social symptoms. And so I think there is a lot of overlap even within these three domains, as they call.
mhm, mhm. And schizophrenia sort of famously has an onset in early adulthood. And so, I don't remember all the specific details here, but typically, your early mid 20s would be when someone often starts to manifest the symptoms of schizophrenia. And I've always understood it as probably being tied with either sort of the end of adolescence or the beginning of the quote unquote adult brain being quote unquote fully developed. What do we know about the developmental side of this?
So the 18 to 21 is the average age of onset in men, and 21 to 25 is the average age of onset in women. There are factors, there might be protective factors related to sex hormones, for example, but then there are also societal factors that impact that as well. But I think it is by definition a neurodevelopmental disorder. They're across multiple levels, everything from prenatal infections to insults, developmental insults throughout life, interacting with genetic risks are then what give rise to the schizophrenia. And so I think there's really a solid neurodevelopmental component. And there are theories that, Daniel Weinberg and others have put forward, and even those have been used to then trying to model schizophrenia in animals. And a lot of them will then use these neurodevelopmental insults to introduce circuit dysfunctions that resemble schizophrenia even in animal models.
Mhm. And how much do we know about the biological causes? I mean, I know that there's still much to be discovered here, but are there certain types of circuit deficiencies, certain types of biological dysfunctions, certain networks in the brain that are reliably dysfunctional in a certain direction in most schizophrenics?
Yeah, and so in patients with schizophrenia, you see hallmarks, right? So there's this imbalance between limbic and cortical dopamine function, right? And so that's something that's seen. In addition to that, in brain imaging studies, you see changes in the size of the hippocampus, for example, the anterior hippocampus, and that's one of the more robust sort of meta-analysis findings in the brain. But even at a cellular level, you see differences in synaptic pruning, for example. So one of the strongest hits that we have as far as a genome-wide association study for schizophrenia is actually in the major histocompatibility complex, like
Hmm.
the immune-related function. And that has been suggested that, there is a normal, going back to the neurodevelopmental disorder piece, a normal trajectory of brain development, right? Especially when you're young, your brain's trying to understand the world around it, and all of these branches happen at the level of your neurons, right? And then as you grow up, that refinement starts to happen where you start to prune away at some of those branches and to get to that, what is like the fully formed adult brain, if there's such a thing. But, by 25, maybe you've gotten to some place. Your brain still remains plastic. Your brain is still able to create new cells of all sorts, right? So there's still room for change. It's not like, at 25, that's when your brain stops, but maybe that maturation process stops by then. And so one of the things that we do know is that there is aberrant synaptic pruning in schizophrenia as well. And
Mhm.
that might be one way in which that circuit dysfunction manifests itself through neurodevelopment.
Yeah. So roughly speaking, as you develop after birth and you mature, as you become an adult, throughout that whole period, there's phases of synaptic pruning that happen. So you have many more connections to begin with. Many of those actually go away. And part of a big piece of brain maturation is actually the loss or the pruning away of excess connections and the preservation of some.
And
And
the
what
important ones, let's
yeah.
say.
Yeah. And so you're saying that there seems to be a pruning issue in people who develop schizophrenia, and this is actually tied to genes of the immune system.
Yeah. So, and I think there's some really nice, compelling evidence in that. But then there's other genes as well, right? Like we have, it is a polygenic disorder. Right? We have, 80 different de novo mutations. And we have something that's actually called disrupted in schizophrenia 1, a DISC1 gene. And there are genes in catecholamine methyltransferase genes and the dopamine B2 receptor genes and dopamine metabolizing and catabolizing genes. So there is, like, it's a complex disorder and it cannot come down to one gene, but it's that complex interplay between all those genetic factors, and then add on top of that the environment. So
Mhm, mhm.
things like immigration status is a risk factor. Cannabis use, especially during adolescence, is a risk factor. And again, even those relationships are bidirectional, and we'll probably talk about it later as well, especially this relationship between cannabis and schizophrenia.
Yeah.
But I think it remains a complex disorder that we shouldn't try to reduce down to one thing causing it.
Yeah. And so one of the networks or neuromodulatory systems that is tied to schizophrenia and psychosis more generally is the dopamine system.
Yes.
And, I've covered this a lot on the podcast in different ways. People often talk about the dopamine system or the reward system or the mesolimbic dopamine reward system. And as hopefully many of my listeners have learned at this point, it's too simplistic to think about dopamine as the same as pleasure. Dopamine does not equal pleasure or reward. It's involved in reward learning, but it's involved in motivation and cognition more generally.
It's involved in locomotion.
Yeah, it's involved in
Right?
locomotion.
We're much simpler than reward
Yeah,
cognition might be too complex. It's even that's why Parkinson's disease
yeah,
is dopaminergic in its etiology.
yeah. And then, maybe one point of reference right here that could be useful to people is that what we call antipsychotic medications that can treat certain types of symptoms for things like schizophrenia well, but not others. They're often acting on the dopamine system. Can you just talk about what we know there a little
Yeah.
bit?
So most antipsychotics, most typical and atypical antipsychotics are modulators of dopamine function, specifically dopamine D2 receptor function in the brain.
Mm-hmm.
They're antagonists of the receptor. And so the older classic typical antipsychotics like haloperidol and others were potent inhibitors of the receptor. But with that comes, again, talking about locomotion, the extrapyramidal symptoms that were there. So that would affect gait, for example, and in the people using antipsychotics. And so that was a problem. And so that's why we've shifted from typical antipsychotics to more atypical antipsychotics. And the difference is really how potent are they as an antagonist at the dopamine D2 receptor and how broad their function is beyond the dopamine D2 receptor. And so things like clozapine, olanzapine, aripiprazole, quetiapine, these are the newer antipsychotics that are atypical, and they are usually associated with less of a potent antagonism at the dopamine D2 receptor, but also they have action at other receptors like the muscarinic and the serotonergic system in the brain. So serotonin versus muscarinic acetylcholinergic system, even histaminergic systems. And so they have then a broader, messier profile, but does not have the same extrapyramidal symptoms associated with the typical antipsychotics.
Mm-hmm. Mm-hmm. And so the dopamine system is gone awry in certain ways in many forms of psychosis, including schizophrenia. What are the antipsychotics typically good at treating, and what are they typically poor at treating when it comes to schizophrenia?
Most antipsychotics are pretty good at treating positive symptoms.
So hallucinations and things like that.
Yes. Not a lot of antipsychotics are all too great at treating negative symptoms.
Mm,
And a lot of them don't even touch cognitive symptoms.
I see.
And my favorite drug, all classes, remains an antipsychotic. It's clozapine, which is probably the most efficacious antipsychotic we have, but because of its black label warning and the side effect profile, it's only used in the most treatment resistant of patients, but it is in fact the most effective antipsychotic we have. It does a pretty good job with positive symptoms, actually can even touch negative symptoms, at least in terms of anhedonia and stuff. It may have some effects on cognition as well.
I see. So the movie A Beautiful Mind with Russell Crowe, where he plays a schizophrenic mathematician, it seems like it was probably actually a pretty good representation of how the treatments typically affect you. So it was taking care of his hallucinations, the positive symptoms, but it left him sort of anhedonic and unmotivated, and sort of it had cognitive side effects as well.
Yeah, no, and that remains a challenge for patients with schizophrenia in general, and then the other thing that often also complicates things, right? Like there's already all sorts of reasons for treatment non-compliance, the paranoia, mistrust of the medical enterprise, and all of that is there. That contributes enough to treatment non-compliance. But add on top of that some of the side effects that come from the antipsychotic medications themselves. Excessive weight gain, metabolic disorder, and all of these other things then also further worsen that treatment non-compliance piece.
Yeah, and I mean that's an important thing to emphasize. Is even though this is a mental disorder, even though these drugs are acting on dopamine, which we think of as a neurotransmitter in the brain doing brain stuff, very, very often with these things, these psychiatric medications, you often have metabolic issues that span the entire body. So these things aren't sort of limited to the linguistic domains that allow us to talk about them.
And those effects are probably central as well as peripheral, right? And
Mhm.
so this is the thing with the brain. Anything that's going to act in one part of the brain is also going to act on the other part of the brain, and that part of the brain is connected to the other part of the brain, right? So, and that's where some of the problems start.
Mhm. So as we mentioned, schizophrenia typically you see the onset in young adulthood, late adolescence, your early 20s, basically. And famously, you hear this often that schizophrenia can be precipitated by some type of stressor at that time period in your life. People often talk about, if you have a genetic predisposition, say to schizophrenia, or it runs in your family, you should avoid psychedelics because they can trigger schizophrenia to emerge, or adolescent use of THC can trigger schizophrenia to emerge in someone with a predisposition. To what extent is that true, and what do we actually know there about these precipitating factors?
So we can think about precipitation in different ways. So let's talk about, on one side, substance-induced psychosis.
Mhm.
That's a real thing. You see it with cannabis. You see it with hallucinogens, psychedelics. You see it with methamphetamine. And when you have substance-induced psychosis, there's usually three paths that happen after that.
And before you go on, so when you say substance-induced psychosis, are you talking about the onset of permanent schizophrenia, or do you mean the onset of a temporary and reversible
And this
psychotic
is an
state?
acute psychotic state induced by substances. And that's where I'm going to get into what the potential outcomes are. In the first case, you actually do end up going from having an acute psychotic state to an organic psychotic
Mhm.
disorder.
So an acute psychosis precipitated by drug use can sometimes turn into a long-lasting
Yeah.
psychosis.
The second one is that you stop using the drug completely and you never have a psychotic episode again. And that's where, there's some sort of level of self-selection as well that happens, right? And
Mm.
so, even with cannabis, where most people will be like, well, I never experienced such a thing, or I've never had a psychotic episode, or I've never felt, even though THC is a psychotomimetic, people are like, I've never experienced such a thing. Well, it's because the person that tried it for the first time and had a bad trip never tried it again.
Yeah, yeah. The person who
Right? And
takes
so,
the edible and has
yeah,
a temporary acute psychotic
yeah,
like state
yeah,
probably doesn't try it again.
try it again, right?
Yeah.
So there's a selection bias that creeps in when people generally talk about this, right? And that's why nothing polarizes people more than cannabis, whether
Right,
cannabis
right.
causes schizophrenia or not, whether it's a
Oh
psychotomimetic
yeah.
or not.
Oh, I mean, I'm sure we'll get into this, but
Yeah,
as someone who worked in the marijuana industry, I mean, yeah, this is just a huge issue. You've got one pocket of people that think cannabis equals psychosis,
yeah,
and you've got another pocket of people that says cannabis can't possibly cause psychosis because
yeah.
I enjoy it.
Oh, and that, but also they'll say, oh, well, cannabis has gone up in potency. Why haven't we seen an increase in the
Yeah.
the prevalence of schizophrenia? And those are all real, valid concerns and questions. Like, I actually tend to find myself somewhere in the middle
Yeah.
of
I do
both
too.
those sections. And I think we need more people in the middle. And I have colleagues that who are preclinical scientists who'll swear that THC causes schizophrenia. And on the other side, I also have preclinical colleagues who are like, THC does not cause schizophrenia, right? And I think the truth is still somewhere in the middle. And I think it's important to have that nuance when we talk about it.
Yeah.
So going back to the thing from the sort of three paths. So one is you develop an organic psychotic disorder. The other one is you never go back to it again. And the third one is anytime you use the substance again, you do have that acute substance-induced psychosis, but you don't have a
Oh,
chronic psychotic disorder.
I see. So, okay. So let me summarize for people
Yeah.
because this is important. And I
Yeah.
rarely hear people sort of bucket things this way. So there are a variety of drugs that are associated with the induction of psychotic states of different kinds. The big ones that often come up the most are methamphetamine, hallucinogens, including certain psychedelics, and things like THC. And there are sort of three ways that psychosis can be induced. One it's temporarily induced, but then that temporary acute psychosis transitions into a chronic form of psychosis, such as schizophrenia in some cases. The second thing that can happen is there's a temporary psychosis, but then when the drug goes away, so does the psychosis. So it's just sort of a side effect of the drug in real time. And then the third is that someone will have that temporary psychosis every single time they try a substance, whereas someone else might just have it once if they try a lot, but they don't have it every other time they try it.
Yeah, so that basically captures all of that. And I think, and so where you then see it in terms of precipitating, and even when this relationship between cannabis use and schizophrenia, where there's probably real signal is in two places: prevalence and age of onset. So there's a greater prevalence of schizophrenia in those that use cannabis during adolescence, from the Swedish conscript studies and others, and there's a lower age of onset. And so, especially with higher THC or more potent forms of cannabis, you do have an earlier age of onset of schizophrenia. So I think that those are the sort of places where you can see it show up.
Yeah. I want to unpack the relationship between cannabis and schizophrenia a little bit further. I want to take a minute to sort of summarize what I understand to be sort of the state of the literature on this and why perhaps there's a lot of confusion and antagonism between people on this point. So the question is, does cannabis use have a relationship with schizophrenia? Is it causally related to the onset of schizophrenia? And, you know, there's questions here related to onset of use. Do you start using it earlier in life versus later? Potency, are you using a relatively potent or less potent form of cannabis, et cetera, et cetera? My sort of bird's eye view of the literature here is, okay, if we just look at the correlation between cannabis use and schizophrenia, there obviously is a correlation there. Nobody disputes that. The question then becomes, okay, is this a correlation that represents a causal linkage, or is this a correlation without causation issue? And then there's a bunch of studies. There's a bunch of correlational epidemiology style studies. There's a bunch of twin studies out there, and they can try to control for as many confounders as possible. And some of them do that, and they come out saying, yes, indeed, there does seem to be some relationship that could be causal between early adolescent use of THC and onset of schizophrenia. And then there's other studies, twin studies, that control for various confounders, including genetics by the nature of a twin study. And they say, actually, when you control for the right confounders, there is no relationship. This is probably correlation, but not causation. I suspect that, as you mentioned earlier, this is an issue where the answer is somewhere in the middle, and there probably are genetic and environmental factors that mean that there is a causal linkage for a subset of the population, but perhaps not for everyone. What is sort of your take on the causal linkage between adolescent THC use and schizophrenia, and what the literature says in totality?
Yeah, so if it's okay with you, I'll get into a little bit of causation versus correlation, a little bit where that comes from and what that technically means, and then we can get into the cannabis and schizophrenia a little bit.
Yeah.
Okay. Yeah. So there's this Hill 1965 Hill's conditions for causation, right? And so some of the things that they talk about in it, does an agent cause a toxicological response? And it was in the context of tobacco smoke. And so the 7 conditions, and there might be more, but they are the strength of the association, so whether there's a relationship between the independent and the dependent variable, the consistency of findings, the biological gradient, is there a dose response? As the dose goes up, as the amount of THC gets higher, do you see this temporal sequence, which comes first, the chicken or the egg, or the cannabis or the schizophrenia? Then there are the biological and the theoretical plausibility and the mechanisms of action, and then coherence with established knowledge. And so there's no other competing hypothesis that might explain it. And then lastly, the specificity of association. So how closely the cause is linked with the outcome. Right? So those are, this is an OG causation correlation criteria that Hill laid out. And so I think going through each of these kind of will help us figure out that piece a little bit.
Yeah,
and so,
there's multiple boxes to check here, and we want to see how many does THC
how many of the boxes can we check, right? Yeah. So then if you look at, maybe there's 12 studies, Arsenio 2002, Tian 1990, Zamet 2011, McGrath 2010, and they all sort of show cannabis exposure and then odds ratio for schizophrenia. And odds ratio for schizophrenia varies, and it's anywhere between two as the amount of cannabis exposure goes up. The odds ratio is anywhere between 2 to 7, and so there is the logistic regression puts the odds ratio across as if you did a meta-analysis across all of these studies around 4. And so, yes, there is a strength of association that's there. Biological plausibility. You take Cyril D'Souza's work where they gave intravenous THC to healthy participants and to those with schizophrenia. In both the healthy participants and in those with schizophrenia, it increases what's called the PANS, the positive and negative symptom score, which is used to assess schizophrenia, right? So it increases it, and it's time dependent. You see increases in positive symptoms showing up around 10 minutes. They last for an hour, and then by 3 or 4 hours, they're gone.
And just to be clear here for people, so positive symptoms, that would include things like paranoia. So if you think people are talking about you, even though you're not literally hallucinating voices, that's a positive symptom. Obviously, some people get that when they consume a lot of THC, and schizophrenics do as well.
But yeah, no, I agree with you completely. But that's just one symptom, right? But the scores that these healthy participants got up to were like 10, 11. So multiple positive symptoms and multiple negative symptoms showed up. So it wasn't just paranoia that showed up.
Right. And I mean, the other thing I wanted to point out there for people too is when we think about something like hallucinations, people often will think about like the visual hallucinations you'll get on a tryptamine psychedelic. Now, if you take something like THC, you're not going to trip in that way. It's not a psychedelic, but very often people will think they hear people talking about them in the next room, and that is a kind of auditory hallucination.
Yeah. So I agree completely, and that makes sense. The other one, and this is where it gets interesting, right? We talked about it earlier. The specificity of association. When one goes up, does the other. And so while there's a paper by Keel et al. That talks about this considerably, and they said that they found no increase in the number of cases of schizophrenia. Thus there is not even an increase in the incidence of schizophrenia to explain. While cannabis use has gone up, it might not be the case. But there are some more recent studies, especially the Swedish, they do a much better job of capturing some of these things, right? And so as far as incidence of schizophrenia per 100,000 is concerned, you've actually seen ebbs and flows, but in the 1970s, it was around 20%. It dropped down to around 15% in the 1980s, and now in the 2000s it was around 30% or 30%, and then it's up close above 30, not 30%, sorry, I didn't mean 30%, it's in 100,000. So 30 in 100,000 versus 35 in 100,000. So, but because it's such small numbers, right, that flutter doesn't show up. It might be 1/4% or 1/2%, and so that's where I think we probably don't see it as well. But I think the where it gets really interesting is genetics. So and this is one of my favorite papers, Pasman 2018. I think, did I send it? Maybe I didn't send it to you. Pasman 2018. It was the title of it is GWAS of Lifetime Cannabis Use Reveals New Risk Loci, Genetic Overlap with Psychiatric Traits, and a Causal Influence of Schizophrenia. So remember, this is a genome-wide association study for lifetime cannabis use. This is not a schizophrenia study.
Okay.
Whether or not you have used lifetime cannabis use is not the most specific outcome, right? It's rather vague. It's whether or not you've used it. So a 1 or a 0. Did you use it?
Have you ever used it or not?
Have you ever used it or not? So, but a lot of the population would fit under a cannabis user in that condition. When you looked at the genetics, so this was 23andMe and UK Biobank, the largest possible data set that we had at that time. Some cool genes showed up. Some of those genes we're studying in the lab right now. They showed that there is genetic overlap with psychopathology generally, but a
Mm.
causal influence of schizophrenia genetics on whether you're going to use lifetime cannabis or not, whether you're going to try cannabis even once in your lifetime.
How do you establish a causal link in a study like that?
So this is what's called Mendelian randomization. Our germline DNA does not change, right? Like no matter what you use, no matter what your environmental influences, our germline DNA stays the same. Right? You have epigenetic changes, but the DNA stays the same. And so what you can do is kind of look at, okay, these are the genes that confer risk for schizophrenia. These are the genes that confer risk for lifetime cannabis use. If you were to look for the genes that confer risk for schizophrenia in the lifetime cannabis use GWAS, how many of those show up there versus the other way around? And so you can get a unidirectionality or bidirectionality. And so this is, you're basically randomizing. By chance, you would have expected a randomization of a Mendelian trait in one population versus another. But
Yes.
one is selectively overrepresented in one population,
It
then you can,
suggests the
yeah,
plausibility that
follow
it's driving it.
it's driving, right? So
Okay.
that's where the word causality is coming from there. And so Mendelian randomization is a wonderful technique, and I think
Yeah. So
I
basically,
think,
yeah, if I'm hearing you correctly, maybe one way to think about this for people is so I'm going to imagine three buckets of studies. One is a purely associational study. So you're
yeah,
simply asking, is one thing statistically associated with another thing? The opposite sort of extreme would be a cause and effect experiment that you would do in like a laboratory animal. You can inject something into an animal or take something away and really look at cause and effect directly. And these Mendelian randomization studies that you're talking about are sort of in the middle in a sense where
yeah.
you can look to see if there is a directionality to the relationship. Do these genes always or nearly always or more than often than chance predict the presence of something else, in this
Yeah.
case, lifetime incidence of schizophrenia, but you don't see it in the other direction. And so it's not merely association, but it's also not a functional test.
Yeah, it's not a function test, and
Yeah.
but there are other studies that have done, cannabis use disorder GWASs, and actually also show, an increased risk for schizophrenia even in that. And so, but then you also see it there's a lot of overlap in traits, right? Like cannabis, you also see higher rates of smoking, and you also see higher rates of alcohol use, and those also overlap with
Right. Right.
schizophrenia. So it could be possibly explained by a third gene or some sort of pleiotropy at the genetic level, right? Like where
Yeah.
it's now the same gene is contributing to multiple outcomes. And so, but it at the very least, it suggests that it couldn't possibly be as causal unidirectional relationship that we make it out to be, right? So that's on the genetic front, but I think also what's important is the high rates of cannabis use disorder in people with schizophrenia already, and so they're already predisposed to cannabis use in some way, shape, or form. But also, I think it's important to recognize that some of these things, like even though that piece that I mentioned about temporal relationship, right? Like cannabis, teens use cannabis. And schizophrenia happens when you're 18 at the very least, if not later. And so just because one came before the other does not mean that that one thing
Right.
caused the other.
Right.
It's just the time that those things
Uh,
happen.
yeah,
It's
right. So
like,
schizophrenia typically has an onset in your 20s,
yeah,
and teens typically do all sorts of stuff,
yeah,
including smoke
all sorts of,
marijuana.
yeah.
Yeah. Yeah.
So, and then even those animal studies that you mentioned, right? Like, so somebody might come and say that, oh, THC causes schizophrenia in rats, right? They also were giving a honking big dose of schizophrenia, of THC injected into an animal. And so that point that I made earlier about Cyril D'Souza's study, that was an intravenous THC. And yes, at an intravenous dose where it's 100% bioavailability, it will be a psychotomimetic. And then if you do that in an animal and give a honking big dose and you inject them 3 times a day, and you keep escalating the dose, and then you assess some, you put them in an open field and you put them whatever
Mhm.
measures that people come up with to say it's schizophrenia, maybe it does cause schizophrenia,
Mhm.
right? But it's not representative of what use looks like. And so in
Yeah.
our hands, for example, when we give vaporized cannabis every day to an adolescent animal, resulting in plasma levels of THC that are much lower, like a 10 to 50-fold lower than what you might give with an injection, you don't see that shift in sensory motor gating in adulthood.
Hmm.
Yes, you do still see changes in cognitive function and stuff, but the hallmark of schizophrenia you don't see anymore. So I think it's important that we are nuanced in even how much weight we put on the animal studies that have happened.
Yeah.
But importantly, I think it's really important that we recognize that that genetic bidirectional relationship makes it very complex. So
Yeah.
because the people that were going to go on to have a risk for schizophrenia anyways were more likely to try out cannabis and possibly develop a cannabis use disorder to begin with.
Hmm. Hmm. And do we know why that might be fundamentally? Is it because things like the dopaminergic circuitry that is
Yeah,
involved in schizophrenia is also involved in things like motivation reward learning, the
yeah,
very circuits that THC interacts with?
yeah. But also, let's think about like all the potential hypotheses, right? Like there's a shared susceptibility hypothesis, what you mentioned, and this could be a genetic susceptibility, like we've been talking about, or it could be a circuit level susceptibility. It could be self-medication, as it's called.
Hmm.
And even though self-medication hypothesis doesn't necessarily hold true, so Kantian was this like psychoanalyst that first suggested it, right, in the context of depression, and then it's been sort of taken and applied to it. You don't, not all of it holds up, right? Like if treatment non-compliance is already an issue in schizophrenia, why would you use cannabis if you don't use your antipsychotics? The symptoms usually arise or the cannabis use usually arises way before the symptoms ever come in. But also, you don't see a relationship between amount of cannabis use and symptoms. In fact, people who have the most severe negative symptoms are in fact protected against substance use.
Hmm.
Right. So self-medication doesn't always check out, but that doesn't mean that somebody who watches some podcast or some YouTube video or some friend somewhere who said, when I felt that, I tried some pot and it made me feel good. And so you're going to try it, even if it actually doesn't medicate you. You're going to be like, all right, let's try it. And, cannabis tends, especially CBD, ends up having like the best PR, right? Like, and so, we can talk about that later as well. The expectancy bias is real. And that's
Right.
why a
Right.
lot of things with cannabis clinical trials never come out of the wash because it's like, the placebo group also shows a considerable effect. And so
Yeah.
that doesn't mean that people aren't in effect using it as self-medication, even if it is in fact associated with worsened outcomes for them through its continued use.
Hmm. Hmm.
Right. So I think there's multiple factors that contribute to it.
Hmm. So, so in your view, after looking at all of the literature on this, if someone just asked you, a regular person just asked you point blank, okay, does cannabis use during adolescence potentially make you more likely to develop schizophrenia? Is there a clear yes or no answer to that question?
I say there is a link. There is a link, and therefore, if there is any chance that you have any genetic risk for schizophrenia, it's better to avoid it. Or start low and go slow and stay low for as long as you can. And like, wait until you're 25, like all of those things, like just delay it. Just enough that sort of cross that scary
Mm-hmm.
period.
Mm-hmm.
But I would never say that cannabis
Yeah.
causes schizophrenia.
So, we talked about this age of onset issue with schizophrenia, and that certain things can precipitate the onset of symptoms. Is there so is it when people develop schizophrenia in early adulthood, is that a sensitive period in the sense that are there people that have all of the hallmarks of a schizophrenia predisposition, a family history, they have the right genes that put them at risk? If you just sort of wait long enough, if you get past the age of 25 or 30, do your chances of developing it sort of go away?
Yes, sometimes you can see late onset schizophrenia, but that it's true. It usually just arises within that range. And what's interesting is even that sex difference that you were mentioning earlier and that we talked about, those who use cannabis, so women who have an earlier onset of schizophrenia often use cannabis. So it can even take away some of that protective effect, for example, or that delay that usually comes. You see it there. But I think it's also important to consider that even when you think about a family relative, a first degree sibling, for example, a first degree relative of somebody who has schizophrenia that does not have schizophrenia, you actually see much higher rates of cannabis use in that population as well.
So if I have someone in my immediate family with schizophrenia and I don't have schizophrenia, even if I never develop it, I'm more
Yeah,
likely to use probably drugs in general,
yeah,
but
yeah,
cannabis in particular.
yeah. Yeah. So
Wow.
I think that again, making this picture that much more complex.
Mm-hmm. I want to talk a little bit about sex differences in the brain here. And this ties into both schizophrenia and cannabis use and how cannabis and THC in particular affects males versus females. Let's start with schizophrenia. My understanding is it's more common in males than females. Is that true?
Yes, and the co-occurrence with the substance use disorder is also more common. So it's male dominant. 75% of dual diagnosis cases are males.
Mm-hmm. How much more common exactly is schizophrenia in males than females? Is it a huge difference?
It's not a huge difference, especially again, it's because it's 1%, right? So
Yeah.
however you slice this, it's going to be a like 60-40, right? Like
Yeah.
you're
Yeah.
not going to get a big differential there.
Mm-hmm. And do we know why that kind of sex bias exists?
I think it's important to then realize how societal expectations, but also um, help-seeking behavior, but also how society views mental illness, and so there's all sorts of layers that complicate this, and then if you add things like race on top of that, it gets like I don't know how if you heard of have you heard of the book Protest Psychosis or the prevalence, the high prevalence of schizophrenia in black males? In fact, it's actually been referred to as the black male disorder, and even an over diagnosis of schizophrenia in black males, or
Mm.
right. And so there's all sorts of societal factors that also affect whether or not somebody is going to be diagnosed with schizophrenia or not.
Right. And I would imagine, when they do a lot of these association studies and these GWAS studies, these twin studies and stuff, if there is a systematic relationship between the rates of schizophrenia and things like sex, males versus females, and things like race, and what sort of lineage you belong to, and it's only 1% of the population, if you have to sort of keep subslicing the groups like this,
Yeah.
the statistical power goes away quite quickly.
And to be honest, we haven't done a great job of having diverse data sets
Yeah.
either.
Yeah.
Like what we know most about in neuroscience is the white male brain, white male adult healthy brains, right? Like that's what we have studied the most. And then every sort of additional cut that you add just gets it further and further lower. So even when it comes to genetic studies, we don't have a lot of genetic studies in populations outside European Americans and then African Americans. That would be the second. And then beyond that, we don't have much at
Mhm.
all. And so
Mhm.
even the genetic associations that I'm telling you about today, they only are in one racial group or two, maybe.
Yeah.
So
Um, One thing that I know that there is a decent amount known is the effects of THC in males versus females. Can you start to talk about that a little bit? And like, give us a bird's eye view to start with. How does THC impact men versus women differently? Is it more potent for one of the sexes than the other?
yeah, so I think there's different layers to it, right? So potency, the technical definition when it comes to pharmacology, is how well that molecule, the THC molecule, interacts with the receptor in the brain that is a part of your endocannabinoid system, but interacts with these exogenous cannabinoids. And that would be the CB1 receptor, for example, the cannabinoid receptor 1. Right. So there are differences there between males and females, and that's related to both expression level, but also because it's the GPCR, it's a G protein coupled receptor, all of those downstream mechanisms that are
Mhm.
activated, they also have sex differences. And
But
so that's the
yeah,
pharmacodynamics
but
of it.
the key molecular component that is responsible for the psychoactive effects of THC, the CB1 receptor, right out of the gate, tends to vary systematically in terms of where it's expressed and how much is expressed in males versus females.
Yeah, and there are differences. They're not huge differences, but there are differences there. But importantly, it's not just how the drug interacts with the brain. The other piece of it is how much drug gets into the body and how that drug is distributed in the body and how your body gets rid of the drug. And that's the pharmacokinetics piece. And that's just as important, if not more important.
I see. Because even if everything in the brain was identical between males and females, they had the exact same CB1 receptor profile, they consumed the exact same dose, you're saying one person's body might clear it more quickly than the other, for example.
Yeah, or metabolize it from that THC compound to another compound, which is, for example, the 11-hydroxy THC compound, which is also an active drug. It's a partial agonist at the CB1 receptor, just like the parent. And women make more of the 11-hydroxy THC compared to men. And so when it comes to edibles, not only might they have differential effects from the parent, but because they make more of the 11-hydroxy THC, which is a longer lasting metabolite in the body, that effect may last even longer, especially with an edible that goes through significant first pass effect, goes through the liver, and a lot of it gets metabolized.
I see. So when it comes to 11-hydroxy THC, the metabolite of THC, how much of it is inside of someone depends on how they consumed it, whether they ate the THC versus, inhaled it, and whether they're male or female.
Yeah. And so those are important factors. But then there are additional layers to it as well. In women, we know of the telescoping effect. And so while they might not use as much, they end up progressing through the stages of the addiction cycle faster than men. In addition to that, we know that there are differences in how quickly men develop tolerance to the effects of THC, which is a very important part of THC of cannabis and THC action that we maybe don't see as much with other drugs. And so the tolerance, the women become more tolerant to THC faster than men do, for example. And so it's at multiple layers, and it's again important. And so even the studies which then give THC to male and female rodents, or they will just give the same dose to both sexes, and then say, males are more likely to have schizophrenia. Well, they were processed very differently, and they did very different things. And I think that's why it's really important to be considerate of the multiple places that each of these compounds can be had.
Yeah. Well, let's think about a 2x2 grid here. So males, females, and then inhaled THC and edible THC. If we were to give everyone the same exact dose of inhaled or edible THC, males and females, the exact same dose, and they all have the exact same use history, which quadrant of the square is going to have the strongest psychoactive effects? Would it be the females eating THC?
Depends on what time you're asking. Right? Like how many hours after taking it, right? Because that peak, right? Like so what you might call a visual analog scale measure of how high are you, right? Like you just ask somebody to draw on a line from 0 to 10, how high are you when they draw it? That already scales, right? Like, so you see, I'm going to try to draw a graph with my hands, X and Y axis. Time after taking it, the peak is higher, plasma concentration is higher when it comes to inhaled THC, and that peak might be higher. And then it comes down quickly. Whereas that one doesn't get up to the same way, but then lasts a whole lot longer. So are you looking at area under the curve, how long that effect lasts, or what that peak height is, and then on top of that, add the differences between what you might call volume of distribution, right? Like more fat in the body versus less fat in the body versus women. So then THC quickly, even though it enters into your bloodstream, gets redistributed to those fatty tissues. And then it slowly leaches out over time. And so then it's possible that somebody who has more fat tissue, it goes there quickly, and then your plasma level may not be as high, but there's enough of it stored in your fat tissue. And then you're going to keep feeling that effect. You're going to keep testing positive on a urine test. You're going to, so there's, this is why
what Well, instead of thinking about blood levels,
Yeah,
what if we just have a behavioral measure of intoxication?
yeah. But
How,
then
how,
tolerance. Are you saying this is a first time user,
yeah,
never
let's
used
say
it?
everyone's first time.
Okay. See, so that's where this is where like people, all of these companies, especially when I was first starting the lab in Canada, legalized cannabis, they were coming to me like, we want to establish a roadside test for cannabis.
Hmm.
I was like, herein lies the problem, right? Like, there's this really cool thing called the cannabis reverse hysteresis effect. It sounds like a cool name, but again, looking at a graph, and so on the y-axis is exactly what you're mentioning, right? Visual analog score how high do you feel?
Yep.
And on the y-axis is plasma THC concentration. So
Yep.
now, you actually see a circular graph. So when the plasma concentrations have come down again, your VAS still stays high. And so that's
Mm.
why it's really hard to then. So even when you're setting up the question, you said, let's
Yep.
ignore plasma,
So in
let's
other words,
go
you're
with the
saying
high.
when people report that they feel the highest, it's not when their blood levels are the highest.
No, well, there might be a point where it is the highest in both cases,
Yep.
but there are points when the blood levels have come down and you're still feeling really high.
Yep.
And there are other points when you were feeling just as high and your plasma levels were actually higher. And
I
so
see.
the plasma levels are not indicative of how high you feel.
Right. Right. And I guess the natural interpretation of that would be that when your blood levels are high, the drug is, circulating at high levels. It's probably binding to CB1 receptors and things like that. But even after it goes down, there can be long lasting effects that have already been triggered that linger
Yes.
beyond the blood levels.
But also because of the redistribution, right? So it's
Mm.
already gone into the fat, but also because your brain is pretty fat heavy, your brain may have a very different amount of THC than your plasma.
Yes. So, if you get a blood draw to measure your
Yeah,
blood THC levels, it's, venous blood from your
yeah,
arm. That
yeah,
might not be indicative of what the brain blood levels of THC are.
yeah, yeah. Yeah.
Hmm.
So that's where I'm not trying to avoid your question, but I think and it'll also differ on which subjective effects you're talking about.
Mhm.
Euphoria versus sleepiness versus impulsivity, and there's even within it there's something called the Nesbitt's paradox, right? Where you have less locomotion, but you have more impulsivity, and so you end up wanting to do more, but able to do less. Right, so
Mm.
so I think there's depending on which of those subjective effects you think about, it will differ based on males and
Mhm.
females.
Mhm. How common is it for there to be uh, fairly substantial, measurable differences in the reaction that males versus females have to psychotic drugs or psychoactive drugs generally?
Again, they differ both on the pharmacokinetics and pharmacodynamics. And
Mhm.
again, it's really important that we consider that. And that's why, what might be considered an average drink for a male is different from an average drink for a female, or the
Right,
number
right.
of drinks allowed for a male versus female is different. And so all of those factors play into each psychoactive drug with their individual nuance in receptor expression, metabolic enzyme expression, volume of distribution. Is it a fat soluble drug or a water soluble drug? Water soluble drug might get cleared out faster in one sex versus the other. So I think there are clear differences in across psychoactive drugs in how male and females receive them.
Mhm. Are there any genetic differences that affect people's sensitivity to the psychoactive effects of things like THC? For example, I don't know, do people have different alleles of liver enzymes that metabolize it differently?
Yeah, we'll see. It depends on who you ask, right? Like there's like companies that have popped up that basically will be like, do you want to know how you would respond to cannabis? We will genotype you for these four genes, and that we think, that captures all of. And so CYP2C19 or CYP2C11, which is the liver enzyme that metabolizes THC, they'll genotype that, and they might genotype a catecholaminomethyltransferase gene, and there's a gene that we're working on in the lab called CADM2, cell adhesion molecule 2. It's one of the only hits that's repeatable in GWAS studies for lifetime cannabis use. In our own work, we've also looked at it with frequency of cannabis use. And so it's probably contributing, and we've now started to model it in animals and see that it does affect how the pharmacodynamics of THC in that animal model. But what's important is that it's not just one gene. One gene is not going to impact. It's not like warfarin, right? Like where you
Yeah.
can do like point of care pharmacogenetic testing. You're gonna bleed out versus it's not gonna do anything to you. Or codeine. Codeine is the other one. Codeine, I call it a choose your own adventure drug, right? So codeine is a prodrug that's metabolized by CYP2D6 to heroin. And so when your dentist gives you codeine for a wisdom tooth removal, somebody could be high as a kite and have all the analgesia in the world, and that would be what's called an ultra rapid metabolizer or an extensive metabolizer. And then on the other side, you have a poor metabolizer. He's popping all and especially in the Caucasian population, you actually see both ultra rapid metabolizers and poor metabolizers.
Wow.
And so, really, like it's a choose your own adventure
Yeah.
drug.
So for a drug like codeine, there are genetic differences that affect enzyme metabolism of
Yeah,
this drug, such
yeah,
that one person takes codeine, they're essentially getting high on heroin, and another
yeah.
person, it's not very effective at all.
Yeah, because it stays at the parent.
Mhm,
But
mhm.
and that's because it's going from an inactive parent to an active
Yeah.
metabolite. In the case of THC, you're going from an active parent to an active metabolite. So no gene is going to set you off in one
Yeah.
direction or another.
Mhm, mhm. Interesting. So you mentioned something earlier. You mentioned that you've done studies more recently with inhaled THC in animals, whereas classically, going back years and years, there's the majority of studies in rodents and experimental animals usually involve injecting THC into the animal's body, and this is going to lead to very different levels of THC and how long they last and all of that. So it's become more common fairly recently for people to do things like inhaled THC vapor in rodent studies. Can you give us a sense for how those studies where you're looking at inhaled THC have started to change the picture of what we think of in terms of what this drug does?
So I'll start off with it's not perfect by any means. And
Mm-hmm.
I'm by no means saying that the people doing the injected THC studies are not doing something right. We're all trying to, model a uniquely human phenomenon and experience in our animals in some way, shape, or form. And so we're like, if it walks like a duck, talks like a duck, vapes like a duck, then maybe it's not a bad model for the duck, right? But we're all going to miss something in one way or another. For example, THC, when injected, produces very different plasma levels of THC, but also that metabolism is very different, and it's going to differ based on how it's injected as well versus inhaled. But with the one problem with inhaled routes is the animal actually still has volition. With
Right.
an injection, the entire amount is getting in.
Yeah. You can't control if
With
one mouse is breathing 12 times
Yeah,
versus 14
yeah,
times or yeah.
and then so you'll see some of them will actually go and stick their nose right where the port is, and another one's gonna go to the other side and stick their nose in the bedding so they don't inhale as much of it,
Yeah.
right?
Yeah.
So that differs. How much they breathe, how deeply they breathe, how much their body weight is, body fat, all of that. So that really changes things. And so then you need to then have an anchor, something like you're measuring plasma level. You're measuring a behavioral outcome. With cannabinoids, it's easy. There's something called a cannabinoid tetrad. It's a telltale sign. You look at their body temperature, you look at how much they're moving around, you look at how much motor rigidity or catalepsy they have, and you look at anti-nociception, how quickly they perceive a painful stimulus. And that tells you if you have cannabinoid action. Anything that acts as an agonist at the CB1 receptor shows this classic tetrad. And so then you need to be able to anchor it to something real like plasma or brain levels and some behavioral outcome to know how much of it is affecting. So this is just to give you that no way of doing it is perfect, and I'm not advocating for one over another. I'm just saying that both of these, we should be remembering the caveats when it comes to drawing conclusions from these studies. Right? But inhaled has its benefits. So, for example, for the longest time, and it's not for the lack of trying, we have 50, 60 years of people trying to get rodents to self-administer cannabis
Mm-hmm.
intravenously, and we haven't been able to do that. The only model that we've been able to have that in is a squirrel monkey. Squirrel monkeys will self-administer intravenously THC. But when you give it via vaporized route, the animals will self-administer it.
Mm.
When you inject THC into an animal and you look at what's called conditioned place preference, do they want to spend more time in the room that they got the THC or in another room where they didn't get the THC? And you always saw a conditioned place aversion with injected THC, but when you gave it via inhaled vapor, you saw preference.
Hmm.
In fact, in our own studies, like with jewel vapor, with nicotine vapor from a e-cigarette, at doses, at plasma levels that are like in the 100 to 200 nanograms per mil, and if you ever gave that to a rodent in an injected form, they would not they would be gasping for air and find it really aversive. When given inhaled via e-cigarette vapor, they showed conditioned place preference.
Hmm.
Right? So the route of administration makes a difference and it matters and it affects how much they like it, but also how much of it they'll take and how much of it will then get into the brain and then have its effects and what those temporal dynamics are going to look like also change, right? Like inhaled through the lungs is one of the fastest way, aside from IV, of getting something into the brain, right? But along the way, the animals are obligate nose breathers, and we breathe through depending on how stuffed up we are, we can breathe through our mouth and through our nose, and then you can do a French inhale where you let it out through your nose and take it back through your mouth, and you can do all sorts of ways of smoking cannabis. But then people will raise concerns like, well, the animals are taking it in through the mouth, or it's getting deposited in their nasal cavity, and that's not how humans take it. And so there are like, I will be the first to tell you all the problems in the way I do things. It's just a problem when other people don't. Right? I think we need to have more humility with our science, but
Mm.
also not be so dogmatic. Like my way is the right way of doing it, and your way is the wrong way of doing it. And that is not if the listeners get something out of this, is that's they're injectable versus vapor. There's no one right or wrong way. These are all paths to that equal final goal of trying to understand what these drugs do on the brain and body better.
Mm-hmm, mm-hmm. Yes, it's a red flag if someone can't articulate the shortcomings or their method. What about, I want to ask you a little bit more about nicotine vapor. So you've been experimenting with that in animals and stuff. What do we know? Can you just, let's again start at a very high level. What do we know about the differences in terms of the drug effects between vaporized nicotine versus smoked nicotine?
Yeah. So I probably am not giving you a straightforward answer for anything, and I'm sorry for that. But various factors come into play. Cigarettes are tobacco that's been cured, that has a certain amount of nicotine, but it has all of those other things, tobacco-specific nitrosamines, and also things that FDA asks the cigarette manufacturers to put in. Accelerants to make the cigarette burn. Irritants to make it feel irritating when you take too deep of a puff of it, right? So that's that. And so you're limited to the amount of nicotine that the plant can produce that can be cured and then to convert it into a cigarette. E-cigarettes, synthetic nicotine in a propylene glycol vegetable glycerine mixture is not the same and does not have the same limits. But also, you can then change the form of nicotine and change the salt that's there. So a benzoate salt will now make nicotine more likely to cross biological membranes, right? And that's why you see some differences in how cigars are versus cigarettes are. And it's because the salts end up being different and the ionization ends up being different. And so nicotine can cross things better or worse depending on. So e-cigarettes give you a lot of room to experiment. And so that's why when something like Juul comes out with a 59 nanograms per mil, 59 milligrams per mil, or a uh, 5%, as they called it, of Juul e-liquid, that at that time became the highest possible nicotine level that you had in any e-cigarette. That changed the game because everybody else in the e-cigarettes were selling 6 milligrams per milliliter, or up to 30 milligrams per milliliter. Then they also changed the salt that was in, and so that also significantly changed how much of the nicotine would get into the body. And so some of the work we've done, like even a 10-minute exposure to a Juul e-cigarette vapor produces massive levels of nicotine in the plasma and in the brain. Right, and higher than, and even they've done studies where they take a Marlboro Red versus a Juul e-cigarette and an anesthetized animal, and they're just pumping right into the nose, and the levels of nicotine are much higher with the Juul e-cigarette than the Marlboro Red. And so that, I think it's again important how the product is, but then the other part that becomes important is smoking topography, right? With the indoor smoking bans, it meant that maybe every hour, whatever your workplace allowed, wherever you were, you took a smoke break. You had 1 unit of a cigarette, you had a pack a day, 20 packs, whatever, that was the unit. Vaping changes the game when it comes to it. You don't have a unit, right? Like you don't have a cigarette that you're going to smoke. You're not going to light up another one before you come back in, right? Like that was the cigarette. You had your smoke break, you're going back in. You can hit the vape as many times, however many times, discreetly, without the scent, without all of that, wherever you are. And so, and any behavior that you repeat becomes habitual.
Mm-hmm.
Right? And the more often you repeat it, the more contexts you repeat it in. Right? Like cigarette might be a context-specific activity. You have to step outside and you're only in that context using it. Vaping changes that, right? Like I've had schools contact me and ask me to come talk about vaping at their school because they're having plumbing problems because of how often the kids, middle schoolers, are vaping in the bathroom and then flushing it down the toilet. Right? So there's I think there is a lot of difference in the ways that, and there's the perceived safety as well, right? Like, but cigarettes are a low bar, right? Like nothing's worse than cigarettes. It's a single largest preventable cause of death worldwide. So anything would be safer, right? Like probably the exhaust fumes on the car are safer.
Yeah. But I guess to step back for a second, one of the features of the e-cigarettes that's used to market them is that, well, you're not getting all of the nasty combustion byproducts and carcinogens that you find in smoke, and you can titrate the dose better, et cetera, et cetera. But what you're basically saying is the e-cigarettes are very capable of getting your blood levels of nicotine higher, and because of the form factor, your frequency and pattern of consumption is probably going to go up.
Yeah. And what you see is that most people end up being dual users anyway. So even the ones that were using it to quit smoking combustible tobacco end up becoming dual users. Now they're using their cigarettes and they're also hitting the vape, so that the troughs so you're basically, with cigarettes, you were playing this peak and trough game, right? Like nicotine levels drop, you bring it up for a little bit, then they drop again, and then you bring it up. So you're trying to maintain it in that sweet spot throughout the day where your nicotinic acetylcholine receptors are maximally occupied by nicotine, right? Now you can sort of hit it in the middle and get a little bit of that nicotine hit. And that's so now most people end up being dual users. And the problem is the people that were going to be never users in the first place, that were never going to take on cigarette smoking, but now vaping ends up bringing them there.
What about, so I want to ask you about the e-cigarettes, in particular, the propylene glycol, the other stuff in these e-cigarette fluids besides the nicotine. What do we know about those things? Are they problematic in any way? Are they relatively inert?
We haven't had a long enough runtime with them. Propylene glycol and vegetable glycerin, yes, with those posters that you find that it can produce dangerous chemicals, and it's true when you heat propylene glycol or vegetable glycerin up to a certain point, it can produce some number of harmful chemicals. Yes, that number is still much lower than the harmful chemicals in cigarette smoke. And yes, as time goes on, we might see effects overall. And yes, it affects lung volume. And yes, it might increase or change risk for emphysema. And yes, it impacts, lung function and surfactant amount and all of that. Like that is all true. But we can't draw a straight line between that and lung cancer like we could with tobacco-specific nitrosamines.
Mhm.
But the other thing that also is important to consider with this is that it may actually have rewarding effects of its own as well.
What do you mean?
Animals will readily self-administer propylene glycol.
Oh, propylene glycol without
Yeah,
any nicotine.
yeah, right. It's not inert in
Ah,
the way we think
so
about it.
propylene glycol vapor, animals will self-administer that.
Yeah, yeah. And then
Huh.
you add flavors on top of that, flavors that interact with, and we already know with menthol and nicotine and how that interaction happened and how that affects it. But then other flavorants, how they interact, both at the level of just the fact that it's a flavor and it's going to have its effects on making it more likely that you use it, but also how it interacts with the pharmacology of nicotine. So I think there's multiple sort of layers in how that gets affected.
Hmm. Hmm. What are some of the active projects in the lab right now?
Yeah, so I think one of the things that we're doing. So one goal that we always have in the lab and this is probably why I can state Hill's causation points off the top of my head, is to try to get at causality, right? Take things from human genome-wide association studies or human imaging studies, reverse translate them into the animal models, and begin to get at the causal influence of whatever the factor is, right? Like if a study is showing that vaping changes brain function and connectivity in this way, in a human study. For example, they're doing the ABCD study, 10,000 12-year-olds that they've recruited. They're scanning them every 2 years. And so there's data, real nice data coming out from it from a large data set. But again, there are compounds and it remains associational, right? So what we're doing is actually taking animal models where we're exposing animals to vaping or nicotine or cannabis or edibles for that matter, or even have a model, an animal model right now of accidental cannabis edible overdose. Because that's another thing that we've seen since legalization has happened is kids, pets, even older adults will eat too much of a pack of gummies or a chocolate bar, and then end up in the ER with acute cannabis poisoning. And so we have an animal model of acute cannabis poisoning that we're trying to both establish ways of reducing the harm or impact, but also seeing what are those long-term consequences of an acute edible poisoning incident. But then on the reverse translation side, we're taking findings from human neuroimaging studies, trying to bring them back into the animal models by using similar methods of exposure and similar methods of studying the brain. So using preclinical MR imaging, magnetic resonance imaging, functional connectivity, and structural connectivity, we can then see, okay, that's what the signal was in the humans. This is what the signal is in an animal. So this component of it is probably causally related. And just like with the neuroimaging studies, we're also reverse translating findings from genome-wide association studies, like that 2018 Pasman paper. There's a gene there called the CADM2, the cell adhesion molecule 2 gene. And so we took those findings, brought it into an animal model. We have an animal model where that gene is knocked out. And now we can begin to see if that gene is no longer there, how do these animals react to THC or cannabis? How do they like it? Do they not like it? And to again begin to establish causality. That gene does causally contribute to risk for cannabis use. And in addition to that, we're doing a lot of studies looking at social drug use behavior. So for a long time, studies that have done drug intake or drug exposure end up singly housing animals, and that can be a confound by itself. When no, aside from the people that drink by themselves in their living room, not a lot of substance use in humans happens in your lonesome, right? So we've established methods where animals can be together and they can drink at the same time, and you can know exactly how much each animal is drinking in a social context. And then we can look at whether if one of them drinks more than the others, how does that affect the others? Or if one of them also is exposed to cannabis or uses or exposed to nicotine at the same time, how does their drinking change versus how does the drinking of everybody around them change? How does stress affect it? So those are some of the, in a 30-second whirlwind tour of the type of work that we're doing.
I also want to ask you a general question about animal models
Yeah.
for human mental illness. So, depending on your perspective, you can get very different answers to a question like this. Um, SSRIs were developed in the 80s and the early 90s. We don't really have new versions of SSRIs. Yeah, I mean, there's some new ones, but they're all just sort of still the first generation SSRIs, and it's sort of the best we can do in terms of giving people a pill to help with depression. As you know, right, most of the antipsychotics I think they've been around for decades and decades. We don't really have new ones that have been developed, and they work for the positive
Except for
symptoms.
Cobenfy, the
Yeah,
one that FDA just approved.
well, yeah, we can come to that.
Yeah.
And they work for the positive symptoms of schizophrenia, but they don't work for many other aspects of psychosis. And then, of course, if you just think about the difference between a human being and a mouse or a rat or whatever, you can, of course, model these things in rodents to some extent because we're all mammals. We all have neurons in our brain. We all have dopamine receptors and so forth. But at the end of the day, how well can you model something like schizophrenia in a rodent? They don't have language. They don't have the types of complex cognition we do. And those are very much at the core of what's disrupted in a human schizophrenic. So what are sort of the limits of these animal models in your view?
I think you captured a lot of them, right? The best model for something is the same, like the best model for a cat is the same cat, or at least preferably a cat, right? That remains the case. These conditions are uniquely human conditions with all sorts of social, genetic, developmental, social determinants of health. All of these factors impact the milieu in which these conditions happen. Right. And so I think it's important that we recognize that and we again have humility around the models that we do use. But there are ways to try to bridge that translational gap that exists as well. In fact, the drugs that you mentioned are actually evidence of the success of rodent models. Antipsychotics, antidepressants, ADHD medications, even things that are used for varenicline for cigarette smoking. Like all of these came from rodent models and they work and we've gotten them to a place where we are able to use them in patients. So I think that by itself is a great testament to the utility of the models, but then also a cautionary note on the limitations of those models as well. So when you mentioned SSRIs, we've established that these were the SSRIs, and then we established some behaviors, sort of forced swim test and tail suspension test, and right, like maybe elevated plus maze, right? And these were like, oh my gosh, these are these predictive valid assays that tell you whether an antidepressant is going to work or not. And then we spent our whole life trying to come up with Me Too drugs, the drugs that are similar to the other drugs, and just testing them on these. And somehow we convinced ourselves that a mouse or a rat swimming in a vat of water was a model for depression.
Mm-hmm.
And that's where it became problematic, right? And so while it had utility and some screening ability for a drug, that was not depression. And so I think we need to keep questioning ourselves with any models that we have. whether there is theoretical validity. Is it based on a theoretical basis of that condition? Is there predictive validity? Does a drug that work in humans also work in alleviating whatever it is in this model? And then the third one is face validity, with whether it looks like, you're not usually putting somebody who has depression into a swimming pool and asking them to swim. Right. So that right. So that does it look does the anhedonia look like what it looks like in humans? And so I think those are the three, but I think I'd add a fourth dimension to it, something I call diagnostic validity. I don't know what others call it, or um, that if the way you study it in humans and then you try to study it in animals in the same way, it could be with MRI, it could be with EEG, it could be with biomarkers, it could be, and those also look the same, then you're one step closer. Right? And then you can try to bridge that gap by using techniques that are used in both. And then adding the techniques that you can do in animals, but also mimicking the routes of administration a little bit. And so I think that is one place that you can get to. But I think an important other place that is really important to consider is, yeah, and we started with this, and I think maybe coming back to it at the end is important as well. All of those overlaps that we mentioned between the diagnostic criteria for DSM also apply in animals, right? Like behavior for an animal, it's most of the times it's going to do something, it's going to be through its movement, right? And so its locomotion is going to affect how we perceive its cognition, or how we perceive its positive symptoms, or how we perceive its negative symptoms. And so I think stepping back from having a model of, I have a rat model of schizophrenia, depression, bipolar disorder, whatever. Instead, we can have, in this model, we are testing this transdiagnostic construct of impulsivity. In this model, we're testing the transdiagnostic construct of anhedonia, which shows up in depression, which shows up in bipolar disorder, which shows up in schizophrenia. And so that then also de-risks it a little bit, because you're not going to have one model of a disease that's going to capture all facets of it. And then you can use genetic tools, right? Like where you can then take findings from genome-wide association studies, bring them into animal models, or use the genetic diversity that exists in an outbred rodent population, for example. And then you can see these are the animals that are the most impulsive, and these are the animals that are the least impulsive. What are the differences between those two? And so I think there are ways of, I think science is evolving enough, but I think we're also, and then, for a large part of neuroscience, we're often studying things that are largely conserved. Breathing circuits, and circuits for thermoregulation, circuits for, right? So I think there's a lot of room and a lot that we haven't figured out about the brain that we still can use rodents and other
Yeah. Yeah, I mean, I guess, when I think about something like schizophrenia and I think about the mesolimbic dopamine system, it's going to hook into lots of other systems, including systems that are highly divergent between rodents and humans, like, parts of the association cortex, the so-called higher parts of the brain. But some of that core dopaminergic circuitry is itself very, very conserved between mammals. So even though it hooks into other things that lead to different manifestations in humans versus rodents, when we talk about different forms of psychosis, say, there still is, to some extent, a common core that's highly evolutionarily conserved.
Yeah. Yeah.
So let's, like, just another general question here that's a little bit more practical for people. Genome sequencing is cheaper than ever. You've got tools out there like 23andMe. You can get your full genome sequence now for relatively cheap. There's different platforms that enable people to do this. Are there cost-effective and easy ways for people to see if they've got genetic risk factors for psychiatric illnesses? And if so, do you recommend those, or are they maybe risky to use? Like, what is your take on using some of these genome sequencing tools to assess one's own risk of psychiatric illness?
Yeah, I think the data governance piece is an important piece there. Um, who owns the data, who owns your genetic data, and
Hmm.
what they do with it, right? Like, are they filing patents on drugs based on your genetic data, right? Or so there, and is it for profit or is it not? And all of that, I think, is an important factor to consider, and it's an important caveat. I personally wouldn't do any direct to consumer um, for myself. I haven't done it, and I haven't done it for my kids, and I wouldn't do it, especially now that one of the big ones is about to go under, and who's going to buy out that data? Where is it going to end up? What is it going to get used for? And so those are the important questions there. But I also think it's too simplistic to then be able to say, oh, this one gene, you are a variant in it. This is like, it's genetic determinism, I think, is, or reductionism on that front is one piece, right? Like, there's no guarantee that just because you have a variant in this, that you're going to now go on to develop that, right? And so I think probably the closer thing is if you have a family member who has cannabis-induced psychosis and then develops schizophrenia, then probably exercising caution is more is anchored on something more real than a data-driven approach that's going to be impacted by all sorts of things, right? Like, what are the training data sets that they have? If you're, from some part of the world that they just didn't have a lot of data on, then those genetic determinations may in fact be completely inaccurate,
Yeah, yeah. So like if you
right?
have something like 23andMe and they're saying you've
Yeah,
got this allele, therefore you've got
yeah,
this risk,
yeah,
it may or may not apply to you as an individual based on the training data set that led them to put that element in their
yeah,
UI.
yeah,
Yeah. Interesting. I mean, basically it sounds like what you're saying is, yeah, I mean, you can get genome sequencing done, you can buy 23andMe or whatever, and you can get, data that is
yeah,
about you and your genetic risk
yeah,
profile. But at the end of the day, it's not that much more informative than knowing who your family risk history.
yeah. Yeah, and I feel like we are starting to lean on one way more over the other. We don't know people anymore. We don't know family members anymore. We don't talk to each other anymore. But we put a lot of weight on a piece of paper that came out that if you think about something like Theranos, maybe some of the data isn't even based on anything.
Yeah, yeah.
Maybe the report they sent back was just completely made up, right? So yeah.
Hmm. We've covered a lot already. Is there anything you want to reiterate or any kind of things you want to tie together from all the topics that we covered today?
I think it's important that we recognize that comorbidity or polysubstance use is the rule and not the exception. And I think as scientists, what I'll make is a case against purity, where like what do we do our clinical trials in? We do test these drugs in unicorns, right? Like, you cannot have any comorbid condition. You can't have a substance use disorder, and you can't have a mental health condition, and you can't have a metabolic disorder, and you can't have. And then we expect it to go out into the real world, into the real population, and then we have to put on a black box warning on the drug because it's associated with increased risk for suicidality. This happened with varenicline, for example. But then when they actually did the test and actually included people who are actually the smokers, 90% of cigarettes are used by 10% of the population, those with serious mental illness, there wasn't any higher risk of suicidality or any other psychiatric complication when they actually tested in a representative population. So
Hmm.
we need to stop testing unicorns and studying unicorns. I think we need to study real people with real conditions, and I guess a case against purity and a case for diversity in our research setups and even in the animal models, right? Like outbred strains, heterogeneous rat strains, like mouse strains. I think there's benefit in embracing messiness and diversity.
All right. Professor Jibran Khokhar, thank you again for your time.
My pleasure.
The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.